Hey guys, so today we are going to be talking about hemostasis, which is like arrest of bleeding. So we'll talk about the various steps included in hemostasis. Firstly, we'll talk about vasoconstriction, which is the initial response, and this is like mediated by substances which are released locally, like serotonin. Then we've got the hemostatic plug formation or the primary plug formation. This is a temporary measure to actually stop the initial bleeding, which is with the help of platelets. Now, that is the first step. Then we have got the coagulation cascade, which will involve the intrinsic and the extrinsic pathways, respectively, which will lead to the formation of the fibrin plug, which is the secondary plug. And then lastly, we'll have the two processes called as clot retraction and fibrinolysis, which will actually lead to the dissolving of the fibrin plug that has been formed at the site of injury once the tissue injury is actually healed. So moving ahead, to understand what happens in the primary plug formation, we need to know a few things. Let's say there are two basic steps which will occur in the formation of the primary plug in case of platelets, which will be the aggregation and the adhesion of the platelets. So the adhesion of the platelet to the actual site of injury will depend on the complex glycoprotein 1b 9 to 5 complex. So this is with the help of 1 Willebrand factor. What will happen is this will help for the platelets to actually adhere to the site of injury. Then there will be a glycoprotein 2b 3a complex which will mediate the aggregation of the platelets. So like a bundle of platelets will be formed at that site to actually cover the entirety of the damaged tissue so as to prevent the bleeding initially. And this will be with the help of binding to fibrinogen. Now this is important because this was a MCQ before. So glycoprotein 2b 3a complex which will bind with fibrinogen to aggregate, not to adhere, but to aggregate the various platelets. Moving ahead, we've got a diagrammatic representation. So now what we need to understand is any coagulation pathway will actually be initiated because of the release of collagen. So this is a normal endothelium which is damaged, and now collagen is released at the site of injury. Now this will lead to the adhesion of platelets. These are platelets with these alpha granules. These green color are actually alpha granules, which will come on to later. Now what will happen is 1 Willebrand factor with the help of the GP2B 3A complex, with the help of GP1B 9 to 5 complex will actually help in the adhesion of the platelet. So here we have 1 Willebrand dependent adhesion, which will include 1B 9 to 5 complex right here. So this is 1 Willebrand factor and this right here is the complex. And there's also one independent process which is independent of 1 Willebrand factor, which will occur with the help of GP1A 2A complex. Now what happens is in 1 Willebrand disease, this complex which only helps in 15% of the total adhesive process will help in the adhesion. So that is why what we'll have at the end is in 1 Willebrand disease the adhesion itself is very less, and that is why aggregation and all the later steps will also be hampered. Moving ahead, we've got aggregation which is going to occur with the help of this complex right here. So let me just zoom in right there. So here we can see that this right here is the fibrinogen and this is the 2B 3A complex which is actually helping in the entire process. Now I have also mentioned about the alpha granules over here, that the last step which the platelets can actually do is cause the activation of the coagulation process. So what platelets will do is they'll release alpha granules. Alpha granules will contain various factors like factor 5, factor 9, which will then factor 11, which will actually help in the activation of the coagulation cascade. Moving on to the coagulation cascade. Now coagulation depends upon the chemical reactions between the 12 vital clotting factors. So we know that there are going to be two pathways. Now the trick is very simple. You need to understand what is extrinsic and intrinsic first. So what intrinsic is suggesting is that the damage is inside the blood vessel or to the blood vessel per se. And extrinsic means that the damage is to the tissue surrounding the blood vessel. So extrinsic damage is going to be the tissue surrounding this blood vessel. Now tissue thromboplastin factor 3. So extrinsic pathway, tissue damage, tissue thromboplastin factor 3. Now what we need to understand again is that the entire aim of this both pathways is to activate factor 10. Like the main aim is to activate factor 10 because factor 10 is commonly then going to activate prothrombin to thrombin, and then the further processes will occur. So the main aim of both these pathways is the activation of factor 10, right? So if factor 3, now I am telling you how to learn about the extrinsic mechanism first. So if factor 3 is activated and factor 10 needs to be activated, you activate the factor 7 because 3 plus 7 is 10. So this is like a trick to remember this. So first we'll have the tissue thromboplastin like factor 3 activation, then we'll have the activation of factor 7, which is the pro-convertin factor, and then we'll have the activation of factor 10, which is the steward power factor. Then prothrombin and thrombin. So before that, intrinsic pathway, the trick is very simple: 12, 11, 9, 10. 12, 11, 9, 10. So 10 is finally. Before that we'll have 12, which is the hegemon factor, which is released because of like the damage to the actual blood vessel, which will activate the factor 11, which will then activate factor 9, which will then activate factor 10. So all this entire step will take place in intrinsic and the extrinsic pathway just to activate the factor 10 finally. Moving ahead, we've got once the factor 10 is activated, we know that factor 10 along with the factor 5 activated factor 5. Now why is this important? Because like if there is any problem with factor 5, then what will happen is that the coagulation cascade is affected to a certain degree. Now moving ahead, factor 10 along with factor 5 and calcium as well, because calcium is also essential for every step after this. Essentially after the activation of factor 10, every step will contain calcium. So prothrombin will activate convert itself into thrombin, which has dual function. Now what I mean by this is like better explained when I tell you about the fibrinolysis, right? So calcium, with the help of calcium, thrombin will activate fibrinogen to form fibrin, and this conversion includes the conversion of two terminal, one central domain of fibrinogen like proteinaceous structure to one central domain. Now once this is formed, what we are getting is not actually fibrin. We are getting small pieces of the thread called as protofibrils. Now these protofibrils will be joining in a number of about 15 to 20 to form a fibrin monomer, not a fibrin polymer. Now factor 13, which is the fibrin stabilizing factor, will actually cross-link the monomers to form a polymer or a proper fibrin polymer, which will actually lead to the fibrin plug formation. So protofibrils are formed, they are joined in the number of 15 to 12. Factor 13 will cause cross-linking and it will form the fibrin polymer. So what we need to understand is factor 13 is also very essential in the last step of the fibrin plug formation. Lastly, we come to a very important concept of clot retraction, because as much as the very step of fibrin clot formation is important, the clot retraction is also very important because it is mediated with the help of platelets and the other components such as thrombin. Now I'll come to the dual function of thrombin over here. Now firstly, clot retraction is not fibrinolysis. Clot retraction is just the formation of thrombus. Now thrombus, right, we can confuse ourselves by looking at the fibrin plug and calling it the thrombus, but thrombus is not fibrin plug. When platelets see that the fibrin plug has been formed and now I need to convert it into a thrombus, then what a platelet will do is it will just extend a filopodia, just like a pseudopodia, like it is a equivalent to pseudopodia. So it will just extend a filopodia and retract the entire clot and take its position, and the fibrin plug will be internalized, right? So it will be internalized, and this right here, this complex which we have got now is the thrombus. So now we have formed the thrombus, and now we are going to lyse the thrombus. So fibrinolysis is going to be the removal of this thrombus once the tissue healing has occurred. Now there is no need of the thrombus or the mass to remain there. So now the fibrinolytic pathway will be activated. Now here I'll explain the dual nature of thrombin. Now what happens is, once the tissue is actually healed, the endothelium is like formed again, right? So there is a new endothelial formation. Now these endothelial cells, they release a local hormone called as thrombomodulin. Now thrombomodulin along with thrombin will activate protein CS complex, which is protein C and protein S, which will activate the plasminogen to convert it into plasmin. So thrombomodulin local hormone, thrombin, which was actually also helping in the fibrin plug formation. It is also helping in the lysis of that plug. So it has a dual function. They will activate protein C and protein S complex, which will then activate the plasminogen to convert it into plasmin, which will lyse the clot. Now the last and a very important point, which is clinically important, is that this plasmin will break down the fibrinogen to give fibrins to give us dimers of fibrin. Now these are small fibrin dimers which can be used, like these are called as D dimers, and D dimer test is done to actually check if the clotting mechanisms are in an orderly fashion, right? So if the clotting mechanisms are working fine, then the D dimer test is suggestive of the activity of the blood clotting mechanism. So, and D dimer was also very frequently done test in during COVID times. So like ferritin tests or D dimer. So these were all the tests which were being done. So one of them is D dimer, and this is an important point clinically because it helps in the checking of the entire cascade, because this is essentially the last step of any clotting mechanism, right from the primary plug formation to the secondary plug formation to the clot retraction to the fibrinolysis. So this is a last step. So this will help you to check the entirety of the mechanism. So that is why this is very clinically very important. And that's it, guys. So the sources for this video were Ganong, Gaitan and GK Pal. So I hope you enjoyed the video. I know I have not uploaded in quite some time, but I will try to increase the frequency. And let me know if you want any particular topic to be explained. I'll try to put out a video about that. Thank you, guys. I hope you enjoy the video.